This video will cover part 2 of subtopic 6.5, Neurons and Synapses. So the understanding, the applications, and skills will be related to synaptic transmission. So just a little bit of background to set the context of what will be covered in this video, in case it wasn't clear in the previous video. So when we're talking about neurons, we're really talking about the nervous system of an organism, and the nervous system is what coordinates the actions of complex organisms via the transmission of electrochemical signals, those neural impulses or action potentials. These signals, these action potentials, are transmitted by a specialized network of cells called neurons, which we talked about in the previous video. So the diagram just kind of shows the main components of the human nervous system and the different organs and cells involved, and it's mostly neurons. So just a quick review then of how neurons are specialized then to transmit these action potentials. So remember, they have a particular function, so they are going to differentiate to allow for this specialized function. And so neurons on their cell body, which look familiar to you as a cell, have these extensions coming off of them called dendrites. The dendrites receive stimulation or a stimulus, and then allow that action potential or nerve impulse to be generated. That nerve impulse is carried down the axon, which is a very long extension from the cell, which ends in these branches called the synaptic or the axon terminals, which will connect to an adjacent cell. So what this video will focus on here is this little area where we have an axon terminal of our presynaptic cell, and then our dendrite membrane, or just the cell membrane on that postsynaptic cell. And then when a nerve impulse reaches the end of the axon, basically there's going to be a release of chemicals that will travel and make contact with this membrane on the adjacent cell that will stimulate the transmission of that nerve impulse in that adjacent cell. And so that cell will carry that signal forward, or if there is a muscle cell here, for example, it will allow that muscle to contract or relax. So in that transmission of signal to other cells, so once that action potential reaches the synaptic terminus of a neuron, so basically the end of the axon, how does that message continue on to the next cell? So there are two ways the signal can be transmitted. So electrically, that action potential simply jumps to the next cell. It is rare, but it is found in the central nervous system. The one that we will focus on in this video is that chemical transmission of signal. So the action potential will trigger the release of a chemical or a class of chemicals called neurotransmitters from the synaptic terminus of one cell that can lead to an action potential in the next cell's dendrites. A cholinergic synapse is a synapse for a neuron that produces a neurotransmitter called acetylcholine, which we'll talk about more in this video, which sends messages to other neurons or skeletal muscle cells. So the neurotransmitter that IB will want you to know specifically about is the neurotransmitter acetylcholine. So we'll look at kind of what sets the context for that synaptic transmission. So in terms of that transfer to the target, or the creation of that synapse, so nerve cells do not make direct contact with one another, but they are separated by what is called a synapse or a synaptic cleft, which is a gap between the axon of one neuron and the dendrite or cell body of the next neuron. So the two cell membranes don't touch directly. They're very close together, but there is a gap between them. So in synapses where there's a muscle cell, that gap is called a neuromuscular junction. And as I stated in the previous slide, the nerve impulses that we will consider do not jump across the synapse, but they are carried by neurotransmitter. So these chemicals will rapidly cross the synapse by diffusion and will affect the state of the membrane potential of the nerve or muscle cell. And so because it affects the state of the membrane potential, this will then trigger some type of action in that cell. So either another action potential or contraction in a muscle. There are many different types of chemicals that have been identified in this area, and then the neurotransmitter, as I mentioned, that we'll focus on is a neurotransmitter that causes muscle contraction, which is acetylcholine. So during synaptic transmission, there are a number of events that occur. So the nerve transmission will reach the presynaptic membrane, basically the end of the axon and then you have depolarization of the presynaptic membrane will cause calcium ions to diffuse through channels in the membrane into the neuron. So there are calcium ions just outside of the membrane, and they will diffuse into the neuron. The calcium that comes into the cell will fuse with these vesicles, or causes vesicles containing the neurotransmitter to move to the presynaptic membrane and fuse with it. The neurotransmitter is released into the synaptic cleft through this process of exocytosis, and then the neurotransmitter diffuses across the membrane, so it's at a high concentration from where it gets released, and then it will rapidly diffuse to an area of low concentration. That area of low concentration is the postsynaptic membrane, where that neurotransmitter will fuse with receptors on that postsynaptic membrane. So the neurotransmitter will bind to a receptor on the postsynaptic membrane, causing adjacent sodium channels to open. The sodium ions will diffuse down their concentration gradient into the postsynaptic neuron, causing the threshold potential to be reached and triggering an action potential. So within that synapse now, you still have neurotransmitter there, and that neurotransmitter is rapidly broken down and removed from the synaptic cleft. So the neurotransmitter in this case that we'll consider is acetylcholine. Acetylcholine only will remain bound to its receptor for a short time. There is an enzyme present in the synaptic cleft called acetylcholinesterase, which will break down acetylcholine into two parts. So it'll break it down into a molecule called choline and another molecule called acetate. The choline gets reabsorbed into the presynaptic neuron, where it is converted back into the active neurotransmitter by recombining it with an acetyl group. So it gets turned back into acetylcholine by combining it with an acetyl group. So diagrammatically, what this all looks like is we have our nerve impulse traveling down the axon. As that nerve impulse travels down the axon, it will cause these calcium channels to open up. So we have high levels of calcium on the extracellular side. This high level of calcium ions on the extracellular side will diffuse rapidly into the cell, and once in the cell, there are these vesicles, these synaptic vesicles, which contain neurotransmitter in them. These are the little red spheres. This calcium now will cause these vesicles to now move to what we call the presynaptic membrane. So if this gap here is our synaptic cleft or our synapse, then the membrane before the synaptic cleft is going to be our presynaptic membrane, and then the membrane located on the other side of the synaptic cleft is going to be our postsynaptic membrane. So now we've had calcium move into the cell. It'll cause these vesicles to move to the presynaptic membrane, fuse with it in that process of exocytosis. The neurotransmitter, in this case acetylcholine, is released into the synapse, where it will now diffuse across the synapse and bind to receptors on the postsynaptic membrane. These receptors will trigger sodium channels to open up, and you'll have sodium starting to move into the postsynaptic cell. This will eventually trigger a nerve impulse or action potential, continuing transmission of that signal. So now in the synaptic cleft, you have acetylcholine that is left over, which needs to be rapidly broken down. So there is an enzyme called acetylcholinesterase that will take acetylcholine, which is here, sorry, gotta get the OH group in there, and will now cleave acetylcholine into two parts. So it'll basically cleave it here so that you get an acetyl group created and choline left over. So this is no longer able to act on the postsynaptic receptors. So choline now will get reabsorbed into the presynaptic membrane, where it'll be turned back into acetylcholine and placed back into additional vesicles, so that the next nerve impulse that comes along will trigger these vesicles again to move to the presynaptic membrane, releasing this newly formed acetylcholine, triggering another nerve impulse in that postsynaptic cell. So that is postsynaptic transmission, or synaptic transmission. So now there are compounds that can prevent synaptic transmission from occurring. There is a certain category of insecticides referred to as neonicotinoids, which are synthetic compounds that are similar in structure to nicotine, that will bind to acetylcholine receptors in the central nervous system of insects. So neonicotinoids will be specific for those acetylcholine receptors in insects. The enzyme within those insects, acetylcholinesterase, their version of it, will not break down the neonicotinoids, and so the binding becomes irreversible. So the neonicotinoids will not be broken down, and so because they now bind to those acetylcholine receptors, they remain bound. So any acetylcholine looking to now trigger synaptic or looking to trigger a nerve impulse will not happen. So this will ultimately prevent synaptic transmission from occurring, which will result in paralysis of those insects and then death. Now, neonicotinoids are very effective insecticides. Farmers really like using them. And mostly because they're very good at getting rid of insects, and they are much less effective on humans and other mammals. So they're not toxic to us, but very toxic to insects. However, there is concern about their effects on honeybees and other beneficial insects. So it's been noted over the years that there's been a very dramatic and very rapid decline in honeybee populations and other pollinating insects as well. And this seems to kind of line up with the use of insecticides such as neonicotinoids. So neonicotinoids, where they get used is they get used in insect pest control and agriculture. So they are water soluble, which allows them to be applied to soil and be taken up by the plants, dissolved in the water and entering the plant roots. So they're very good at killing off all kinds of horrible pests that destroy our crops. But as mentioned in the previous slide, they will kill beneficial insects as well. So the way that these neonicotinoids work is that if this is our situation of normal neurotransmission, so we have acetylcholine being released from that presynaptic membrane, acting on that post-synaptic membrane, you have acetylcholinesterase, which will break down that acetylcholine, allowing the next neurotransmission to occur, or allowing other transmissions to occur. However, when neonicotinoids are present, they will bind irreversibly to that postsynaptic membrane. Acetylcholinesterase does not break down these neonicotinoids, so you can get release of acetylcholine, but acetylcholine doesn't have anywhere to act. So neurotransmission does not occur. And so this will ultimately result in paralysis and death of that insect. So neonicotinoids, as I mentioned, also have unintended effects. So they seem to be affecting honeybee populations and also populations of pollinating insects, which has effects on agriculture, especially agriculture that depends on flowering plants and trees to produce fruits, for example. And so the last slide will deal with research into learning and memory and how all this is kind of figured out. So remember, those neurons are the same neurons that we find in our brain and our nervous system. And those are the same neurons that are going to be involved now in how we learn and how we remember things. So the fields of psychology, chemistry, biology, and medicine, all very different fields, basically all have to combine to contribute or have combined and do collaborate to contribute to our knowledge of memory and learning. So the human brain is complex, and it's kind of considered one of the last great frontiers in biology. It's not well understood how memories are created, how they're stored, how they're retrieved. So that human brain is very complex, which makes research into memory and learning difficult. So oftentimes, complex biological systems are going to be studied using simpler models. You can break things down into very simple components. It allows very basic biology to be figured out, and that can be applied to more complex systems. So biologists will often use invertebrates that have simpler nervous systems as compared to humans. Now, there is a sea snail or a sea slug that is often used because it has very few but very large neurons that can be easily seen. So this picture here on the right shows the neurons within the sea slug. So there's very few of them, they're very big, and they can be easily seen, they can be easily manipulated as well. And so the way the sea snail gets used is that it has a structure called a siphon that it will retract when stimulated or when poked, and it will retract this as a defense mechanism and can actually learn from experience. So stimulating different parts of it, can allow it to learn from experience, and stimulating different parts can figure out which neural pathways get activated in retracting that siphon. So that is the end of this video for part 2 of 6.5, Neurons and Synapses.